we've got a few things I want to cover. We're doing the Cell Respiration Lab and the Photosynthesis Lab together, right? And I'll focus on the Cell Respiration Lab first. Right, it's when aerobic cell respiration means we're using oxygen and we're using things like glucose, which is a carbohydrate, right? That's the CHO is an acronym for carbohydrate. And we're burning it to CO2 and oxygen and water. Right. So we've got a couple experiments that we normally do, you guys would be doing in lab, but instead I'm going to do them up here in the front of the class. And we're using germinating peas as a model to represent cellular respiration. So what is a pea? You know, you cook them and eat them, but they're a seed. You plant these. There's some in this jar. We've got dry peas that they're referred to as being non-germinating because, right, they're not using cellular respiration basically if they're dry. So we think of them as a control when we use this in this experiment, right? They're not respiring, they're not using cellular respiration. Then I've got this jar, right, with germinating peas in them. And I've got them in an enclosed jar. It's all stoppered up. And I've had this going for the past couple hours. Normally you would set this up at the DN lab, let it go for an hour. And what you're looking to do, right, as long as they're germinating, they're converting the glucose in the pea, right? A pea contains starch, right? What is starch composed of? Yeah, glucoses. So what they do when they start to germinate is they convert the starch to glucose. Then glucose goes through the mitochondria in the pea, and you get carbon dioxide produced, and of course ATP. Right? So the ATP is used by the pea to make a little root. So when you look at these germinating peas, there's a little white, right? It's basically a root that, so if this was in the ground, it would start to absorb water from the ground and nutrients. And then eventually it would sprout up, and then it would make a leaf. And then what it would do, right? Then it would use photosynthesis, like all plants do. But until it gets the leaf, right, like something like a pea plant, or any sort of seed that you plant, right, until the leaves are made by the plant, it's got to depend on the starch or molecules like starch that are in the seed. So they store energy that way, or store glucose. And that's why we eat peas, right, because there's starch, or we eat other vegetables because there's starch in them, or we eat fruits. So now what I want to do, if we did this experiment in lab, at some point after, we want to know, are those peas in the jar making carbon dioxide? Okay, so if I bubble if there was carbon dioxide here, we bubble it through this test tube with the water in it, and the other thing we want to add is phenol red. So let's add that now and make sure it's red, because sometimes it's still, yeah, it's red. Good. So if it's red, that means it's above pH 7. That's why the phenol red, right, looks red. I want the camera to see. Now, what happens if I put, if I bubble, or if I put CO2 in the water? What happens to the CO2? It becomes carbonic acid. You know, you say it's dissolved CO2 is another, so when you pop open a can that's carbonated, the CO2, the carbonic acid in the solution or whatever's in the can becomes CO2 gas and it bubbles out. So now what I want to do is add water. What this does is it bubbles what was in this jar, right? It comes out the top and bubbles into the test tube. So the water that was in the test tube, because it's changed to a yellow color, that means that the CO2 produced the carbonic acid. The CO2 came from the cell respiration of the germinating peas. And then the carbonic acid caused the pH to go from 7 to below 7. We don't know exactly how low it is. It's not a strong acid by any means. So that, what we use this jar for is to see production of CO2 as a result of cell respiration. Right, that was the purpose of that jar. Now the volume meter, and then if I bubble water, if I bubble the oxygen through or bubble what was in this jar, there wouldn't be any CO2 in this. It's the negative control. And you would expect the solution to remain red. I'm not going to go through that. Now the volume meter, that's the name of this apparatus. And it has what's called a pipette here. And there are peas in here that are germinating, just like they germinated in we had germinating peas in the mason jar. But they're in these test tubes, right? And this is a closed container. And I put I'm not going to put another one in there, but this morning I put a it's called potassium hydroxide, KOH, pellet. It's a solid. It's a white like little chip. You can't see it because of the cotton, but it's hanging out in there. And so while it's respiring, it's using up the oxygen gas in the tube and then replacing it with CO2. So the volume is going down for the oxygen, but the CO2 counteracts it so that the volume doesn't change. So to actually measure how much oxygen is being consumed, we just have to get rid of the CO2 that's produced. And we can do that by using the KOH pellets. The KOH pellet, potassium hydroxide, absorbs carbon dioxide gas so that we can measure the amount of oxygen that's being consumed. The mason jar measures CO2 production. We're using the volume meter to measure oxygen consumption. And the way we can do that is I put green food coloring. Now, technically we should add the green food coloring to this pipette, and in this tube they're non-germinating, so they're not consuming oxygen, right? But we're just pretend like we wouldn't expect that to move at all. But what happens is this dye, it starts off, say, like at 0.7 or whatever, you mark it, and then as it as the peas that are germinating are using cellular respiration, they're going to use up the oxygen in that tube, and because we're absorbing the CO2, right, that doesn't have any effect, and the volume in the tube is going to get reduced. If we can actually watch the right, the dye will go in this direction in, right, and it will slowly, I can see it moving a little bit right now, but it slowly keeps moving in that direction as the oxygen's consumed. So the volume meter, that's the name of this, the volume meter, meter means it measures volume. We're measuring using this pipette here. It's telling us exactly how many, if we could put it in milliliters. It typically in one lab it might be like 1.9 milliliters of oxygen gas is consumed in about an hour. When the green dye goes from this point to this point, it's 1 mil. And what will happen during the lab is it'll go down to about here. It'll get so far down that you have to reset it. But so the volume meter is a way of measuring oxygen consumption, and we're using germinating peas to represent cellular respiration. Now, when you take the practical, many people, I ask them questions about the germinating peas, and they think they're photosynthesizing. If the pea doesn't have a leaf, it cannot photosynthesize. And then just to recap that concept of what cellular respiration is, it's aerobic because we use oxygen. CO, right, this say glucose is burned to CO2. The germinating peas, right, are a model of cell respiration. We are gonna, at the end of this experiment, it starts off red, but we bubble what was in the gases that were generated in the tube from the germinating peas through this tube by adding water, like I did. And then so this one's only orange, but mine turned a nice yellow. So it worked. But this, on the practical, you might see this image and I'll ask you questions about it. It shouldn't be that difficult, especially if you come to this lab or watch the video. Now what the volume, right, volume meter, that's what you call this apparatus. It's used to measure consumption of CO2 through cellular respiration. They're not photosynthesizing the pea. What are the dry peas used for in both of these experiments? As a negative control. Those peas are not respiring or using cell respiration, right? They are dormant. There's that KOH, potassium hydroxide absorbs CO2. And then we didn't set this one up, but this is referred to as a fermentation tube. When you ferment, you're not necessarily needing oxygen gas to do this. But, so it's not cellular respiration, right? We refer to it as fermenting. But in this fermentation tube, we mix yeast and glucose together, and then they convert the glucose to carbon dioxide. And you can kind of see the yeast at the bottom of the tube. And as they make the CO2, it comes up on this end, right? It comes up over here, but it just escapes out the top. But this sort of, say, kind of traps it. And over time, it goes down. It'll even, I've seen it go all the way down. It just pushes the liquid out.